Okay. So, hello everyone. Good morning to the folks in the US and good evening to everyone here in India. It's our privilege to be talking to you all about science. So, three of us from Bayer Biotech Center in Bangalore, that is myself, Anik, Tillai, and Shobha, will be sharing with you some of our insights that we've got on science, hopefully to inspire you all a little bit about science. Okay, so to get started, I'm wondering whether any of them in the US will be wondering why people up in India are up till midnight to talk to us about science. Well, it's basically just to show you all that we are really inspired by science and what it can do for mankind. So that's why we love to talk about science. So that's why we are here. Okay, so to give you a little background before we get into this, so we were invited to talk about biotechnology. So we thought we'll share with you all some of the basics of biotechnology and how it could be used. Okay, so science, as you all are aware, can do many things, good and bad, right? And a lot of our, I mean, here in our world, we are facing a lot of problems. Okay, one of them could be is our population growth. So we're soon going to have so many people on this earth who we have to feed, and we're not going to have more land to grow enough food to feed these people, right? So we here at Bayer use different approaches to see how we can tackle this problem, how we can kind of produce more from less. So this is where science comes in. So we try and use different facets of science, different domains of science and the scientific knowledge that we have in order to kind of custom make products for farmers around the world. Okay, so here we'll share with you all some of those things, mostly focus on biotechnology and how it can be used. Okay, so without saying anything more, I'll pass on the baton to Thilai, who will take you all through first the basics of molecular biology, the important tool in biotechnology. Okay, so with that, Thilai, over to you.
Yeah, thanks, Anik. Am I audible?
Yes, Thilai, you're very
Yeah,
audible. I can hear
sure.
you.
Yeah,
Yep.
thank you so much. Good morning, everyone, and good evening to many of the people who are taking call in India. So another 20-25 minutes, we will just talk about what is the basics of molecular biology, right? I'm not going into the agenda detail, but again, this is the agenda we are going to follow for today's session, right? Having said that, I will just move on to the next slide. Can you see my second slide? Yeah. So the first topic, what we are going to discuss is on molecular biology. So many people in the meeting today, so you may be aware of what is DNA, what is RNA, what is protein, but I want to just touch base with the basics of the molecular biology. So again, molecular biology is a study of the biology in the molecular level, so which includes interaction between DNA, RNA, and protein. You may be aware about the central dogma process, right? So we will have a look in a very brief manner, and again we will just proceed further with the next topics. So as you all know, every cell, right, has its own components. Like DNA is present inside the nucleus. So if you just take an example of a cell, so cell will have the nucleus, and inside the nucleus we have the chromosome. Again, each new cell nucleus will have the chromosome, and these chromosomes are tightly packed with the DNA. And the DNA is a double helix structure which consists of adenine, thymine, guanine, and cytosine. So the thumb rule is adenine will always pair with thymine, and guanine will always pair with cytosine. So again, coming to different forms of DNA, there are many forms of DNA. One among the form is A form of DNA, and another one is B form, and another one is Z form. We will just have a look of these forms in the later slides. And in the first slide, I was just talking about DNA, RNA, and protein. So see, this is the structure of RNA. So RNA, or sorry, RNA, or a single stranded nature. whereas DNA is a double stranded. So this we call as a double stranded. Those two strands will be there. Again, if you just look into RNA, the nucleotides, what it made with this is cytosine, guanine, adenine, and uracil. But whereas in DNA, this uracil will be replaced with thymine. So DNA will have cytosine, guanine, adenine, and thymine. So that's a difference between RNA and the DNA. And as I said, RNA is a single stranded one, which has only one strand, and DNA will have two strands. And this is the structure of DNA, whereas the nucleotide will have the sugar and the phosphate backbone. And I'm not going to just go through each and every one. There are many fun facts about the DNA. See, you maybe wondered how big the DNA is going to be, right? So again, if we unwind our DNA, again, it's going to be meters and meters together. So that big is that the length of the DNA is such a huge one, and it's been packed very compactly inside the nucleus, and which the information which is present inside the nucleus or inside the DNA, which is going to govern the genotype or phenotype of any particular organism, if it is a plant or human, that is going to design. So again, when coming to the chromosomes, human has 46 chromosomes, that is not 23 pairs of chromosome, of which no, only one pair is sex chromosome, rest all it's in autosomes. Again, these are the ones which decides the autosomes are the ones which going to decide the appearance or nature of an individual, whereas the sex chromosome is going to decide whether it's a male or female. So these are the other examples for the chromosome numbers. Fruit fly has 8 chromosomes. Similarly, again, tobacco, barley, and all the other has different sets of chromosomes. And we just spoke about the cell. Within the cell, we just know that nucleus will be there, and inside the nucleus, all the chromosomes will be there, and now chromosome has the tightly packed DNA. So as I said, in the chromosome or in the DNA, all the characteristics will be coded or mentioned inside the DNA. So how it has been coded? So inside the DNA, there's a lot of genes present over there. So what is a gene? Again, the genes are the ones which decides your, like say for example, if people have a curly hair, so that's the one which is going to decide the trait. So for that curly hair, the gene has to express, or the gene has to be in a dominant way, so that gene the curly hair nature will come to a human. Similarly, there are multiple genes will be present inside the chromosome, and which in fact gives the trait of the individuals. So if you just talk about gene, okay, so gene will be guided, or it will be promoted by a promoter. Again, without the promoter, the gene cannot be in an active form. So all the gene, it should have a promoter and the terminator to just say, or to program the promoter where to stop and where to start. So this will be done by the terminator. So if you just take a gene, so the gene should have promoter and a terminator, right? So it will be just started with the engine like a promoter, and will be terminated by the terminator or 3' UTR. And once the DNA again, DNA has to replicate, and it has to give its progeny, or it has to give its copies. So what will happen is, again, the next step is called transcription. So whereas the DNA will unwind, and with the use of RNA polymerase, the mRNA will get formed. So as I said, RNAs are going to be a single standard one, and again, it will have all the nucleotides. So once the mRNA has been formed, so that mRNA has to be translated to a protein. again, based on the protein availability, the traits or the characters will get determined. So once the mRNA, that mRNA means it's a messenger RNA. See, once mRNA has been formed, again, it will have its own nucleotide, like G, A, U, and C. So it has to form the protein synthesis. So the proteins have been coded with these tables. So based on this combination, so the amino acids will get formed, or the amino acid will get synthesized from the mRNA based on this combination. Say for again, in a given translation, okay, there should be a start. So always this translation start is with ATG. If the gene or if the promoter sees an ATG in the sequence, okay, in the open reading frame or in the coding region, okay, which is meant for a particular gene, so the translation will get started. So the start codon is called, like, 1 minute, I will admit this. The start codon is methionine. So if ATG, if it is there, if it is methionine, it is a start for a translation. Again, now followed by that, the lot of amino acids combination will be formed based on these nucleotides. And if the gene has to be stopped, okay, again, now these are the stop codons: UAG, UAT, and UAA. So these are the stop codons which recognized by the no process, and it will get terminated. And one quick example is, again, you may be surprised what the promoter sequence is going to have. So this is a thumb rule for the promoter sequence. Always, above the ATG or the start of the coding region, the promoter sequence will present. Say upstream to the coding region, -10 region. That's 10 nucleotides downstream of the coding region. There will be a Tata box, that is the characteristic features of the promoter. And again, further 35 nucleotides down, so there will be a cat box, or we call this as a cat box. So the representative nucleotide, it's going to be TTGA-CAT. So these are the representative sequence which should present upstream to the gene. Can you see the example over here? Like, no, this is the example. So like ATG upstream to this is the promoter sequence, which will have the Tata box and the cat box. Hello.
Yeah, Tilai, go on.
Yeah, sure.
Tilai,
Okay.
yeah,
Yeah.
there's one question from Pallavi asking, what is the state of the chromosome in the nucleus? Does it float or something? That's what she's asking.
Okay. So see, during the transcription, it will unwind. Okay. Based on the necessity, based on the information or the signal, okay, it will unwind. Then rest of the time it will be in an intact form. I don't think it will float, but again, it will be arranged inside the nucleus, and when it is required for the transcription, so it unwinds. Does that may answer her question?
Yeah, yes, Tilai, I think it does.
Okay.
Go
See,
ahead.
once the transcription has happened inside the nucleus, the product, again, as we discussed earlier, the mRNA. So mRNA will get formed, and it will just come out from the nucleus, and it will just flows into the cytoplasm, right? So where, again, it gets all the other components for the translation. So again, these are the substance which is needed for the protein synthesis. So one is the mRNA, which has been transcribed from the DNA, right? And another one is the rRNA, ribosomal RNA, and the third one is the transfer RNA. So these three things combine together for the protein synthesis. Again, these process will happen outside the nucleus. So what will happen is the messenger RNA will float inside the cytoplasm, and this rRNA will bind to this messenger RNA and this tRNA, where you are seeing as a kind of what do you call the stamping pad. So it will just bind to the ATG. This is an ATG. So because this is an mRNA, so the thymine has been replaced with a uracil. So ATG, this is a start. This triple letter code stands for methionine, and always the start is with methionine. So once it sees the ATG, the ribosomal RNA, which has been bound here with this major and minor subunits, so along with the transfer DNA, it will start synthesizing the protein. Say once it's been bound over AUG, it will start synthesizing UAG. That is the complementary nucleotide, which has been synthesized by the tRNA. Once this synthesis has happened, then again the tRNA will get moved to the next triplet codes, that is UCC. Then now this protein will have, like say for example, if this UAG, if it is code for isoleucine, so isoleucine is a protein has been synthesized, and it will be waiting for the next protein to form. So similarly, till the end, again the protein will form, and you will get a complete protein from the given gene or for a given gene. So this is the entire process of translation, right? Any doubts in this? Just it's a brief. I've not gone in detail of these basics. Any doubts here? Anik, I'm not seeing the message box. So if there
Yeah,
are any questions in the chat box, please do let me know.
yeah, I'll do that, Tila. Yeah, you can go ahead.
Sure. Okay, so these are the basics of molecular biology. Now we are going to look into how the cloning can be done. So again, say for example, just keep something as an example in mind, so that again you can just follow the subsequent slides. So if you want to clone a drought gene or insect resistant gene, we need to have the target gene. Okay, I know it has to be cloned into our transformation vector. If it is be a plant transformation, or if you want to do any other things, it has to be inside the vector. So if we are planning for that, again, we need to have a series of vector. Again, there are some limitations with the vectors. Say, for example, plasmids or extra chromosomal substance, wherein we can just clone maximum of 8 kb. So beyond 8 kb, if someone wants to clone a drought gene of 10 kb, so plasmid is not a good vector for cloning the gene. So they have to choose another vector, which is called bacteriophage, lambda bacteriophage, whereas the people can clone 9 to 25 kb of DNA. And if it is beyond that, again we have to use cosmids or yak or back. Again, these are the representative examples of the cloning vector. So we will just talk more about the plasmid as a vector, so in the coming slides. So we have designed that we are going to clone a 1 kb, that is a 1,000 base pairs of a drought gene for our experiment. So for that, what we have to do, we have to source the gene. Again, do a PCR, take a plant or any organisms which has the drought resistant gene, and do a PCR, that's a polymerase chain reaction using a primers and probes, primers and enzyme, amplify that product, have that product. So we will just treat this as the amplified product, or this is the drought gene which we are going to clone into the plasmid vector. So we have the amplified product, and we have decided that we are going to use a plasmid vector. To clone this insert, that is amplified product or the drought gene, into the plasmid, we need to have a compatible site for cloning. So again, what we have to do is, there is a biological scissor called restriction enzyme, which recognize the given sequence. Say for example, if I say EcoR1 restriction enzyme, that means that particular enzyme will recognize GAATTC sequence. So I have to digest both the insert and the vector with the enzyme. Then after digestion, I have to ligate. Ligation, again, it's a process of binding both the DNA molecules, like the insert and the vector. So once the ligation is done, again I have to do a series of steps to push that ligated product, push that complete vector or a vector containing my drought gene into the E. coli. So these are the steps we follow in the laboratory. Once a protein, once a plasmid has been cloned, again we have to do a heat shock. So this heat shock will loosen the cell membrane of E. coli, and the cloned product will get inserted into the bacteria, and the in the further subsequent selection on the antibiotic media, the positive clones or the clones which has the gene of interest will grow on the media, and it will get selected. And these are the examples of the clones. If it is a, so again, no, as I said, we have digested this with a restriction enzyme. This can go in either antisense or sense or the antisense orientation. So we need to select the orientation of our interest. So mostly we select an sense orientation. So if it is a sense orientation, this is our right clone, and if it is an antisense, this is not of our choice of clone. So we have to omit this. So how do we do that? So once the colonies are formed, we need to do a isolation of plasmid. So there are many methods in the market, like no commercially available kits are there. But again, this is the traditional method where we can isolate the cloned plasmid from the E. coli. So take the E. coli cell, lies with solutions like sodium hydroxide. So the cell wall will get loosened, and all the components which are present inside the bacteria will come out. You may ask a question, what is going to happen with the bacterial chromosomal DNA? So while we are treating with this sodium hydroxide and other salts, see these bigger chunks of chromosomal DNA will get chopped into bits and pieces, whereas the plasmids are small circular molecules. It will take time to get denatured or broken into pieces. So this will be intact. So within this time frame, what we will do is we will do a brief centrifugation, and the bigger chunks will get settle down at the bottom. We can remove those things. This will contain the cell wall and the chromosomal DNA, whereas the solution, the supernatant, will have the plasmid DNA of our interest. So this plasmid DNA, no, we have to precipitate and take out for doing the QC analysis. So again, the first quality check to say whether it's a clone or non-clone. So we will be performing again. Mostly everyone does the restriction enzyme digest. So again, this is the agarose gel electrophoresis, where the digested. I was just mentioning about the restriction enzyme, right? So take the DNA and digest with the restriction enzyme and see whether you have your insert or not. Say for example, uh, I have taken the DNA and I've digested with eco or one. That's a restriction enzyme. So this is A is a non-clonal one. So if I digest with the restriction enzyme eco or one, it just linearizes. That means this doesn't have any insert over here. Whereas the B, right? Upon digestion with eco R1, it has given a band over here. This is the vector band, and this is the insert band. So this confirms that this is a clone. But this is again, it's a putative clones. Again, if there may be some nucleotide changes or deletions or addition may be there, but it's a putative clone. So scientists, they will select these as a clone. The first round of QC passed plasmid as a clone, and this particular plasmid will be subjected to sequencing for final confirmation. Final confirmation will be done by sequencing. So even though if you're saying it's 100% clone, there should not be any mutation in the plasmid DNA. So once the sequence has been confirmed, then it will be confirmed as a proper clone, and this clone will be taken forward for subsequent analysis. Either it could be for further research, or for plant transformation, or for any other purpose. Any questions here?
There's one question in the chat about, has anyone used
Mhm.
this cloning technology till now to create clones?
Yes, like signed. If you just see a journal again, many people say, as I said, an example, right? Or I'll give you an example of our company. So we have done cloning of this Bacillus thuringiensis gene, Cry1AC, into a plasmid vector. It's been thoroughly characterized. Okay, then it's been taken for plant transformation, and that product, it is available in the market as Bolgard or Bolgard 2. That is a transgenic cotton crop which is there in the market, and it gives the resistance to the cotton pest. People have been using. Any more questions, Anik?
No, Tilay, that's it. That's amazing.
Okay.
She replied, yeah. Tilay, you've got 5 minutes to finish your basics. Okay.
Yeah, I'm done with that, Anik.
Okay.
I will
Yeah.
move on to the
Okay.
next one. Okay.
Good, 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 good. Yeah.
Okay. So we have completed the basics of molecular biology. At the team here, you may be you are about now you know about DNA, RNA, and protein, how it's been made and where it is made, and what is chromosome and what is gene, and what drives the gene, like promoter and the gene of interest and the 3' UTR. And we have learnt about how the cloning will help, or how the clones can be made in a bacteria for further studies, right? The next topic what we are going to discuss today is on the tissue culture and the genetic engineering, right? Again, as you all know, it's a tissue culture. Again, we are going to, for this session, we are going to touch base with plant tissue culture, right? So what is a plant tissue culture? Again, now growing a plant in an artificial, or or not in the soil but inside the laboratory. Okay, so which needs a confined space and a nutrient rich media, wherein the plant cells can be placed over that, and it can be grown into a bigger plants, right? And genetic engineering, okay, is a process. Again, it's a recombinant DNA technology, which we will just look into subsequent slides, how we can alter the gene, or how we can alter the genetic makeup of a given organism, right? These are the two things we are going to see in the next slides. So first, the team should understand what is breeding and what is genetic engineering, right? So as you all know, conventional breeding, we need to have a two parents, and this see, one parent will have a desired trait. So we want to bring that trait to a subsequent generation. So we have to do a crossover here, and that particular character will get into the generation, and this will get multiplied for the next generation. So that is the conventional breeding. Whereas the genetic engineering, what will it does is, again, the part of a gene, whatever, let's say for example, if it is a drought gene, or as I said about our company product, it's a Bacillus thuringiensis isolated gene from Bacillus thuringiensis. Okay, and then clone that into a plasmid vector, and this plasmid vector again do a transformation into the plant and get a trait, okay, or a new trait, that's a biotech trait. That's called the genetic engineering. Again, this conventional breeding and genetic engineering, it's totally a different topic. So we will just see how we do with the genetic engineering stuff. Before moving into genetic engineering or tissue culture, we need to know the basics. As you all know, again, leaves, stems, flowers, whatever, it will be made with a lot of tissues, and each tissues will have thousands and thousands of cell, and within the cell, we'll have a nucleus, okay, and other organelles, right? And within the nucleus, this plant related things will be packed in the chromosome, and it will have the DNA and the genes, right? So plant has a potential, what that means is, again, from a single cell, okay, it can we can just make a complete plant out of a single cell. So whereas other even humans doesn't have that, but it can the similar, say for example, if it is heart tissue, the same heart tissue can be cultured, but not as a human. Whereas in plant, it has a capability to grow from a single cell to a bigger plant. So mm, that is called a totipotency. So we have been using that totipotency to develop a plant from a single cell. So the single cell, whatever we are using, even it's a single cell or any leaf-based explants, we can just give we can make those explant to a generate a complete plant. So we call this as an explant. So now we will just see what are the types of explant. There are many types of explants we use. Again, these explants are starting material for the tissue culture or for genetic engineering or for the tissue culture. Again, where the lab has a well established system for growing from a single cell or a leaf tissue to a higher plants. Say for example, in Bayer we have system for tobacco and rice, corn, cotton, for many other crops. We have well established or well tested tissue culture system for many crops. Again, for the differentiation of the cell, right, it's all based on different plant hormones. One such hormone is auxin, and another hormone is called cytokinin. If we play around with these hormones, we can make we can differentiate the cell either to a shoot or to a root. Okay, so we will just see the next slide. Again, this is the process of tissue culture. So whatever say for example, if it's a carrot plant, if I want to generate another carrot plant, I can take any of the cell, any cell from the carrot plant, okay, and that we call as an explant. Keep that explant in a nutrient rich media, okay, and upon keeping this explant on a nutrient rich media, this will produce a calli. The calli is a undifferentiated mass of cell. Again, just like a cancerous cell, it will just the cell will get proliferated and it gives a mass of cell. So one cell has been formed into thousands and thousands of cell over here. And take this calli or take this individual cell and treat it with different types of hormones. So if based on hormones, say for example, you can see the hormone combination here. If you just put in high cytokinin and low auxin containing media, you will get a shoot development. So take a piece of calli, put it in high cytokinin and low auxin containing media, you will get a shoot development. Once the shoot development has been formed nicely, then cut off the shoot from the explant and put it in a media which contains high auxin and low cytokinin. It will develop a root. So once the root development is done, you will get a complete plant. Take this plant and harden it in a greenhouse. That's again, it's a contained environment, and then take it to the field for further growth. So this is the cycle of tissue culture. We will see it in the next slide. Any doubts here? So this combination is very important. The hormone combination is important to get a calli or shoot or a root development. It's mentioned here. So to get a calli development, you need to have a auxin and cytokinin in equal combination. For root, high auxin, low cytokinin, and for shoot, it's a high cytokinin and low auxin. Again, these are the combinations of hormones which gives maturity, flowering, or fruit development. Say for example, as I said here, for germination, okay, this gibberellin, it's a hormone which helps in germination. But again, for germination, if you use auxin, it is not of any use. Similarly, cytokinin, it won't help. So these are the combinations of hormones which helps in different development stages. Again, if you just think of plant tissue culture, there are many uses of plant tissue culture. It helps in preserving our germplasm, and for doing many modifications of genes and transfer different genes into a plants to bring a, a product out of that. So it has many useful in plant research. Okay, I will just take you through an example of genetic engineering combined with the plant tissue culture, right? We all know how the tissue culture will happen, right? It starts from explant and it will go to callus, and based on the combination of hormones, we will generate a root or a shoot and a complete plant. So now I'll just take you to an example of how the insect resistant plant has been developed in our company. Again, why to create such product? As you all know, I'm going to take cotton as an example. So the yield loss because of this insect attack, it's a huge loss to the farmers. So scientists in the company thought of bringing a resistant product out of that. So what they have done is the strategy is, again, they have introduced a gene from Bacillus thuringiensis. Again, this gene has a capability of killing the cotton pest. So how the mode of action, how the assay works is, this protein, whatever we are cloning, the Bacillus thuringiensis gene, which we are going to put inside the plant, it will produce a protein. This protein will enter into, upon infestation, this protein will get entered into the insect mouth, and the insect mouth will have alkaline pH. This will get solubilized, and it will bound to the receptor, insect receptor, whereas the insect, the further growth or the further things will get arrest over there, and the insect will die. So this is the mode of action for the Bt toxin. That's again, we will just talk about Cry1AC. That's a Cry stands for Bacillus thuringiensis gene. And in our product, we have Bolgard and Bolgard 2 in India. Bolgard is with a single gene, which gives resistance to Indian cotton pest, and Bolgard 2, which gives a broader range of resistance. Right? I will admit this. Okay. So as I said, Bolgard 1, it's a single gene technology. It came to India in 2002. Right, and it gives resistance. Again, it protects, okay, this American bollworm. So if a farmer has Bolgard in their field, if this pest, if it comes and attack this particular transgenic plant, this pest will die. Why? Because again, as I said, mentioned the mode of action in a previous slide. Upon infestation, the worms will, or the insect will get the protein, and the protein, the gut of that particular insect will have the alkaline pH, so the protein will get solubilized and it arrests the growth of the insect, and as a result, the insect dies. Similarly, in 2006, we have launched Bolgard 2, which has two genes, Cry1AC and Cry2AB gene. These are the genes which are present inside the cotton plant, again, which gives the broader range of protection, like bollworms and the Spodoptera, right? So how it helps, how it helps the farmers. So this is the reason, this is the statistics, 2020 statistics, where it says, again, this is the India is exporting cotton. Earlier, India used to import cotton a lot. Now India is in a place where it can export, where it's exporting cotton to other countries. So that's how, again, this technology has made farmers' life to a next level, and farmers are happy. And this reduce the use of pesticides and other stuffs for generating the cotton.
Tillai,
Hello.
Tillai, there is a question.
Yes, Shobha.
How can one obtain DNA of plants? Where it is stored? Like question is it in seeds?
Okay. So DNA can be present in anywhere, even though if you isolate, it can be isolated from leaf, stem, root, anywhere it will present. So as I said, if you just take a cell, okay, DNA will be there. So again, no, cell can be found anywhere in the plants, right? So it will be available throughout the plant. Does that answer?
Yes.
Okay, so I was just explaining about the Bolgard and Bolgard 2 technology, and these are the some of the traits, that's biotech traits, okay, right? These are the transgenic crops which are available in US. Okay, so we have cotton with IR stands for insect resistant and HT stands for herbicide tolerance, right? So this is whatever we have seen in the previous examples, or the insect resistance. So because of the presence of cry1 and cry2 genes, this will give the insect resistance. So insect won't, it can't attack the plant. And similarly for herbicide tolerance, if you spray any herbicides, cotton plant will stay live, whereas the other no the unwanted plants will get killed. So we have no these technologies in cotton and other plants as well. So these transgenic plants are available in US, not in India. In India, we have cotton as transgenic plants. Again, this is on sugar beet, on herbicide tolerance. Similarly, again, papaya for virus resistance. So this is a papaya ring spot virus, whereas if they use this trait, the papaya ring spot virus won't happen in papaya. I will move on to the next slide. Any guess what is this? What is this picture? Okay, I will tell you. This is again, this field, the half of the field which is in brown color, this is all non-BT grown field. That's um, a field without Bolgard or Bolgard 2 technology plants. So there is no infestation, insect infestation was there, again, plants are died, or it doesn't give much of the bowl or the cotton fiber. Whereas the BT field, that's this is the BT field where they've grown the BT technology cotton, where they got a high yield of cotton, right? So this is the difference of difference in the technology. Like this half is a non-BT field and this is the BT field. Okay, Anik, are we on time?
Yes, they're like good, doing good on time. Yep.
Sure. So what next, Anik? I'm done with that.
Are there any questions? Maybe we can take them.
Yeah.
Or there were also, I was thinking there are some questions regarding genetic engineering and tissue culture that had come
Mhm.
up that were posted earlier.
Mhm.
So do you think we can go through some of them?
Yes, Anik. Who is going to project that? Or
I'll just read it out and then
yeah, please, Anik. Yeah.
maybe you'll see. Okay, yeah. So how long does it take to make a genetically modified crop?
Okay. Can I take this question, Anik?
Yes, yes, yes. It's for you, Tilay. Yeah.
Okay, as the team again, now we are talking about that, right? So it starts from the ideation. Okay, so it will take minimum 2 to 3 years for testing in the R&D, and if it has to come to as a product, it will take another 6 to 7 years. So altogether, it will take minimum 10 years. Because from the ideation, it has to go through R&D procedures, again cloning and confirmation, and transformation. That's a tissue culture. Is there any questions? Okay, transformation. Then again, it has to go for multiple rounds of field testing, trials, and other stuff. So minimum 7 to 10 years.
Yeah, thanks, Delay. There's another question. How can we combine biotechnology and astronomy? I don't think we
Hmm.
can combine those two, but anyway, you have any comments? Yeah.
Astro, no, sorry. Um,
Yeah, they are
mm,
completely different fields. So
mhm.
I'm not sure if you could combine those two sciences, but
No,
anyway.
astronomy, if they meant like no, doing a tissue culture or anything in the moon or something like that, there are trials there, but I'm not sure whether no, we are answering for that question.
Correct. Okay. Yeah.
Hmm.
And how can students participate in the biomedical engineering? So you have any comments on biomedical engineering?
See, again, biomedical engineering, it deals with the equipment design and things like that, right?
Correct. Yeah.
Okay, biomedical engineering, that's what I remember and I know. Okay, so how again see now people they want to hear about molecular biology, genetic engineering, markers and other stuff, so they approached us, right? Similarly, again, if they want biomedical engineering, again I feel they have to approach such companies
Okay.
who are into like the equipment design and preparation.
Okay.
Can I add a point here?
Yes,
Yes, Shubha,
Shobha.
please.
Okay, so biomedical engineering is mainly about medicine and engineering. So people who are very passionate about biology and also engineering can take up that biomedical engineering that mainly deals with equipments that are employed in medicine mainly. So you have heard about minimal invasion and the probes that are put into your um, um, I mean, foot track to see what is there, if you have any infection or caused in your stomach, and then maybe your X-ray machine, and then maybe your CT scan machine. All this, how does these machine work? How they are designed? How to operate these machines? And how to analyze the data or the results that come out of these machines or equipments are mainly taught in biomedical engineering. And biotechnology is actually slightly different field where you actually study about biology. Core, the main core subject will be biology in biotechnology, and in biomedical engineering it is like combination of medicine mainly, and then electronics, electronics, robotics, like and mainly in engineering subjects. Does that answer?
I, I think that surely helps, Shobha. Yeah, thanks.
Mhm.
There's another question on is biotechnology a modern science field?
Mhm.
Can I answer that? Yeah.
Yes, Anik.
Okay, so biotechnology actually uh, is not a modern science. Even the whole science of making cheese, making yogurt, making wine is all biotechnology. So biotechnology is basically where you use any living organism to bring about a change in something that we want that benefits us. So mankind has been using it from time immemorial, from many, many thousands of years. But it's off late for the past 30 or 40 years that we've learned different tools that we can use at the molecular level to help speed up these processes. So that's what is kind of gaining momentum now. Okay, there's another question in the chat, Tilai. Which field in biotechnology is giving a promising future?
Mhm. Shobha?
Okay, I can take that. Like based on my knowledge, animal biotechnology, if you study, like there is lot of scope in pharmaceutical industry where they do lot of testing, and nowadays it's getting evolved where people are looking at growing organs in the lab, like say people who are hearing impairment, try to grow that um, um, membrane in the lab, and then implant it or something like that. Lot of experiments are going in that direction. And like drug discovery, where you want to see what drug you need to treat what disease, and all those uh, people who are interested in those things. Pharmaceuticals is a place where you find lot of employment for biotechnology. And specifically agriculture biotechnology is little less compared to employment in agriculture biotechnology is little less compared to pharmaceuticals. And then another field is as we discussed about bio the other one is like uh, this is something new which I also learned recently. So one of my friend was sharing, like there is one field in robotics. Okay, so robotic
Mm.
engineering, like engineers are trying to understand biology, like how a limb is moving, how you move your hand or fingers or eyelids, or how to try, how do you hear something. All this, the people are learning those aspects of biology to design um, robots. So you have, there are a lot of fields now evolving, like you have bioinformatics as well. Like if you are a biologist and you learn biotechnology in depth, there is since the sequencing has become cheap and you have lot of information based on sequencing, there is lot of sense you can make uh, out of those sequencing. So that field as well is evolving. So rather than like how we used to see traditionally, each subject separately, there are a lot of subjects that marry together. Like I said, robotics needs biotechnology or biology. And then even biomedical engineering, where engineers design equipments for medical science people or doctors. There you need biotechnology or biology. And then bioinformatics, data science. There are a lot of fields where you that are getting married, and dual expertise is always a plus point or addition to the students. So people who love multiple subjects can look at studying diverse subjects and then see how you can employ that.
Yeah,
You, if Anik or
yeah,
Pillai
Shobha.
has
Yes,
any
no, no,
inputs, you can add.
no, I think you covered it well. And just for everyone's information, in case you all didn't know, even medicines like insulin and a lot of other vaccines are generated through biotechnology. So that is, they genetically engineer
Mm.
microbes to produce these. So there are a lot of opportunities in industry like that way. Okay, there's one question over here that came up. How can artificial intelligence and biotechnology be integrated? Are there any latest developments or discoveries? Okay, so just to answer this question, I'm sure all of you all are familiar with how data is changing the world, right? Data is also changing the world, even in biotechnology and even in any science fields. In fact, Tillai and Shobha over here are both data scientists, and they were actually scientists in the laboratory who are now moved completely into data science. And how artificial intelligence can be used is this is how they can use historical data to kind of predict experiments in the lab or predict experiments or crosses. So you need not do so many experiments in order to get good results. So there are many other fields like that in artificial intelligence that can be helped to speed up developing newer technologies in biotechnology, be it in crops and in
Mhm.
anything. Okay. So do you all have anything to add to that, Shobha or Deepak? Shobha or
Yeah,
Tilak, how
that's a good
artificial intelligence?
call
Yeah.
out. Yeah, that's a good call out, Nick, again, or to the team again over here. I want to just bring an example over here. See, as I shown again, we have been discussing about the biotech products, right? So it may take 7 to 10 years to come as a product, right? So this all if we just do a work in a traditional way, okay, so starting from the ideation till the product out. But based on the artificial intelligence, what we can do is now we, with the data, with the power of data, what we can just do is we can just predict, we can just develop an algorithm which can predict a gene, whether it will be useful, whether it will be effective, or whether it is going to give a desired trait for us. So with that prediction, we can just minimize couple of years of our research. So that is again, that is a power of AI in biotech research.
Yeah. Okay. Thanks, Tilak. Yeah. And there's another question. As a student in environmental engineering, I want to ask if there's any potential danger in which could result in damaging the environment. Shall I take that, Tilak? Yeah.
Yeah, please.
So the question is whether biotechnology can harm the environment. So it depends on how the technology is used. Now, if the people who are developing the technology have intended to use it in the right way, then it will not damage the environment at all. And moreover, there are a lot of regulators around the globe who are keeping a very strict tab on how research is conducted. In other words, anyone, everyone can't just do any kind of experiments without having these right permits. So it's unless they get these permits, unless a permits are issued, all the people who are working on various aspects of biotechnology work in a manner that can improve the environment or uh, safeguard the environment. And to give you a simple example of the genetically modified crop in India, it is to control a worm, an insect, which would normally be to control it, we would have had to put a lot of insecticide. So now by
Mm.
minimizing the spray of insecticide on the plants, we are actually preventing the unnecessary usage of excessive pesticides. That's saving the environment and the soil and the water. Okay, so that's my answer to that on thing. So these technologies, biotechnology, in fact, can be used to safeguard the environment or to protect the environment. Yeah. So I think Shobha will go on to the next session now because we are right on track. So Shobha, you're ready with
I'll
the thing?
stop.
Yeah.
Yeah.
So Shobha Deepak will be now
Yeah,
presenting
yeah,
and she'll
yes,
take us
Anik.
to, yeah, she'll take us through molecular markers and how the various applications of biotechnology. So we've been talking about biotechnology. She'll also kind of, and you all can keep the questions going and we can discuss about them as and when we take these slide breaks. Okay. Yeah. Yeah, your screen is showing up, Shobha. Yes.
Okay, thank you,
Yeah.
Anik. Okay, um, good morning and good evening, everyone. So, as we heard from Dr. Tilla about basics of biotechnology, we'll continue with some more benefits of this biotechnology knowledge. Okay, so we'll talk something about genetic markers. So I just want to know, like, if I say genetic markers, can anyone say what do you understand from this? Okay, so it is just like something that you can easily identify differences. Say you have you use in your work like two colored pens to mark different things, right? um, Two colored ink, so it is something like that. It's not, there is nothing hi-fi about it, it's like very basic. So you identify the differences using something, and they are called markers. Okay, so in genetics, like we find something where you can easily identify difference between two things which you use in your um, genetics. Okay, there are three major types of markers, if broadly classified. Okay, morphological markers, biochemical markers, and molecular markers. So morphological markers or phenotypic markers. Say like you all have studied Mendelian genetics, right? Where he identified tall plant, and then color, flower color, different flower color in peas and all those. So they what you what you see is phenotypic marker or morphological markers. So variations you find in plant height, color, and horned phenotype in cattle. So in cattle, some get horns, some don't get horns. Those basic things what you see are called are can be used as markers, and they are called morphological markers. And there are something called biochemical markers. So morphological is easily you can see through your eyes. So biochemical markers are not you can not identify with your eyes, so you need some chemicals or equipments or some technique to see the differences. So they example like milk, different kinds, there is some difference in the milk which you can identify using by adding some chemical or some method. You identify, and you see there is there is something which you can identify through urines, and your blood groups. You find AB blood groups, ABO and all blood groups. Those are identified use, they are also biochemical markers. And then milk protein and your serum protein, all these, they are all biochemical markers. So to identify difference between one individual to another individual, either you do some test like blood groups or the serum protein or some of those tests to find the differences in individuals. Okay, and then there is something called molecular markers. So molecular markers are like what are DNA based. So there, so you're not able to easily see and identify the difference. You're not able to identify the differences with some tests. So there is some markers where you find differences on the DNA. So as Tillai was telling, on the DNA, there will be some variations you find from individual to individual. Those variations you use to identify the difference between two individuals or some differences. Okay, so y- so there are many ways where you can identify the differences, but like depending on that limitations, advantage and disadvantage, you choose which method you want to use. Okay, but when you want to see the differences, say morphological marker, if you want to use, so there should be some differences to use, but sometimes you can't identify the differences. So those cases you can't use these small morphological markers, and they don't reflect variability in coding gene. So wherever you see the difference in tall plant and short plant, you can identify, but there is something called something genetically in between that. Okay, so those things you can't identify using morphological markers. And these biochemical markers, as we discussed, like they are age dependent. So early ages, some chemicals are produced, and in later stage of ages, if you consider human beings, some hormones are produced at early age, some hormones in the mid stage of your age, some hormones at your later stage of your age. So there will be variation in that, so you can't use them as biochemical markers. And they are also sex limited, like some are only produced in female, some are produced only in male, those kind. And there are also, they also have influence on environment. Say, like if you're looking for a biochemical marker for drought resistance, that plant which is very happily growing, um, most of the cases you won't see that character of drought resistance expressed using, you can't identify using biochemical markers. So that the any general happy plant, when it is under stress, or where there is less water, or there is where there is harsh sunlight and all, they try to produce some chemicals inside to alter themselves into survive in that environment. So depending on environment, those markers change, so you can't use those biological biochemical markers all the time. Okay, so most preferred one is molecular markers, and it is evolved like this is the most evolving field recently, and all biotechnologists employ molecular markers more than any other markers. So they can detect variation at the coding level, at the DNA level. Okay, coding and non-coding is like, coding is where coding DNA is where the gene is there, and the non-coding region is where the you have the DNA where that doesn't code for any gene. So in that portion also, if there is any difference, you can identify, and you can find high degree of polymorphism. So where there is differences, you can easily identify, you can employ markers to find those differences. Okay, and they are also unaffected by environment, and they are locus specific, means if you identify certain characters, they are always present at that position, so they don't change their position. So if you identify some position, it is easy for you to go back and check or reconfirm that that is present in the same position. Okay, and there are numerous differences throughout the DNA which you can use as molecular markers. So those are the ones that are actually preferred, and people employ those for their advantage. Okay, I'm not a okay. So we'll talk more about this molecular markers. Um, so there are many types of molecular markers. So you find they are called RFLP and RAPD. They are first generation marker, and next evolved is AFLP and SSR SSRs, and then third generation is EST and SNPs. We'll go through all of this individually one by one. So RFLP is a restriction fragment length polymorphism. So you have a long stretch of DNA or DNA where you cut the DNA using restriction enzymes. So we'll see what restriction enzyme is and how do you cut that. You take a DNA and you cut it with some enzymes, and then you see what is the difference in the enzymes. That gives you uh, some hint about differences in two sets of DNA. And then it is you don't need any PCR or hi-fi equipments to do that. That is very basic. Okay, RAPD is random amplified length polymorphism. Here you need PCR, as Tillai was telling about PCR, where you take some you take the whole stretch of DNA and you amplify it. We'll come to all these individually one by one. AFLP is amplified fragment length polymorphism. That is one more technique. SSR is simple sequence repeats. So in DNA, you find some simple sequence repeat. Like if there is a stretch, there would be stretch of 80, 80, 80, 80 continuously many times. So those things are identified, and differences are identified, and you can use them as markers. ESTs are expressed sequence tag. This is mainly with this is mainly used in identification of genes. Will not focus mostly on this. And SNPs is single nucleotide polymorphism. So in a stretch of DNA, if there is difference in one nucleotide also, you can use this for your advantage. Okay. Okay, we'll talk about this one by one. So sorry, RFLP is um, restriction fragment length polymorphism. Okay, what you do here is, so you take out, this is a cell with the DNA, you take out the DNA out of the cell, then what you do is, you amplify that DNA, you multiply that DNA, and then you use some enzymes to cut the DNA. Okay, when you cut the DNA, um, so depending on that enzyme where it is cutting, some DNA will cut here, some DNA will get cut here. So there are many places where the DNA gets cut, and when you run that on the gel, you see different bands like this. Okay, based on that, you see where based on that you identify the polymorphism. So if you take two individuals, so here is an example. Okay, there are if you see round and square and blue, half blue, as parents, and when you do this RFLP for the parents and the siblings, so this is how you get the variation with the siblings. So one will be round blue, half one will be square with half of it blue and white, and another one is completely white square. So those differences you can identify like this. You can see the differences on the gel. Okay, so this same method is can be employed to find out whether you have some disease or somebody is having some disease and all. So what you do is, you take the DNA from two individuals, and then you cut the DNA with an enzyme, and you run on the gel. Okay, so normal person will have will have between normal and disease, most of the DNA fragments are like similar. So but these red ones are where they are dissimilar. Okay, so you have to identify what is the different dissimilar one between these two individual, and then these are the RFLP markers which you can identify for disease detection. So if you see this, if you run a gel for the person with the disease, so if this marker shows up, then you can say this person is having a disease. But if they doesn't show up and you find another two markers, you can say that this person doesn't have this particular disease. That is how you employ this technology to detect diseases or any other thing as well. This is just an example of disease. Okay, moving to the next one. Okay, I talked about restriction enzyme, and Tillai also talked about it. But just to make you understand, I have just put this pictorially. So there are some enzymes in our cells that are called as molecular scissors. They just act like scissors. Okay, one example is that, say there is a sequence here, GAATTC is a DNA sequence, and then what this one of the restriction enzyme that is well known is eco R1. Okay, so what this does is, it needs, wherever this sequence is there, GAATTC, it recognizes this sequence, and what it does is it exactly goes and cuts at G. Okay, so it recognizes all these 6 sequences in the same order it has to be, but while cutting, it cuts at G. Okay, top strand gets cut at this G and bottom strand gets cut at this G. So there will be two pieces of DNA with some extra hanging here. So they are called sticky ends. Okay, these ends can be stuck together, so they are called sticky ends. So these are some of the restriction enzyme example. So you don't have to worry, but just to say that, like, there are many restriction enzyme which has different recognition site depending on where you want to cut. See, this BAM-H1 cuts at GATCC, so it cuts at G. Okay, depending on where you want to have a cut, you can choose your enzymes. Okay, there are something, this is the enzyme that produces sticky end. So there are some enzymes that cuts exactly at the middle, so there is no sticky end produced here, so DNA gets cut at the middle. Okay, so one example is EcoR5. Okay, this EcoR5 enzyme recognizes GATATC, and then it exactly cuts after this GAT. Okay, so you get two fragments with no sticky ends. So these are some restriction enzymes. So how does this RFLP work is, say, there is one mutation here. So in one person, or the person having disease will have prone to disease will have one mutation here. So instead of GATA, he will have GA. Just assume, like, he will have GA disease person, and normal person will have GA TA. So when you put this EcoR5 to cut this, sorry, cut this. So in normal person, this EcoR5 will make a cut here, but in diseased person, it can't make a cut here because it doesn't, the T will not be there. Instead of T, there will be G. So it can't cut here. So depending on that difference, you can identify a normal person and the person who is prone to some disease, or say some drought resistance in case of plants, or some um, something like where you can employ in many places. Okay, um, so okay, we'll go to the next one. Okay, the next marker is RAPD. This is random amplified length polymorphism. So here what you do is you randomly amplify the DNA. You put some primers, as still I said, to amplify the DNA you need some primers. Okay, so you randomly put some primers, and then you amplify the primers, and then you amplify the DNA, sorry, and then you depending on where it is amplified, you get the bands like this, depend on different individuals. So these are 1 2 3 4 are all individuals, and then in each one there is one place where it is getting amplified. So from 2 to 8 9, you can see they are all uniform. So it's getting amplified in one way in all these individuals. So 10 to 12 it is amplifying in different way. So those differences you can identify. Okay, those are called random amplified length polymorphism. And one more thing, the next marker is AFLP, that is amplified fragment length polymorphism, where you should know the sequence of the DNA in the middle. You should be knowing some portion of the sequence of DNA. Then you cut it with some enzymes. Say here they have given example of EcoR1 in one side and PST1 in another side. Depending on its recognition site, it cuts. Okay, then you add something called adapter to that. This is slightly little complicated compared to RAPD and RFLP, but this this also needs little knowledge about the you already have to know the sequence of this green stretch of DNA, then only you can do this, employ this technique. But in RFLP and RAPD, you don't have to know the sequence of DNA, you can randomly do that. Okay, then you put this adapter, and then you identify the difference between um, those sequences. Okay, so coming to simple SSR markers, that is simple sequence repeats. Okay, how does this work is, so on a DNA, in a DNA you find some simple sequence repeats you find. Say stretch of what the example I have given here is CA. You get some full stretch of those CAs. Okay, you identify those regions. Okay, what you do is you employ your primers here. So you put this is the DNA sequence, you add the primers. So in one person there is less number of CA repeats, in another person, second person, there is more number of CA repeats, and in third person there even more number of CAs. So depending on how many CAs are there, when you do amplification and when you run the gel, you see that um, person with a less number of CA, you can identify a band with a blue band. Okay, person with more number of CA will have a red band, will have a band that is at a different position. Okay. Mm, and then person with highest number of CAs will have this band at different position. That's how you identify the differences in the individual. Okay, so these depending on how many repeats are there, it can be 80 sequence also, it can be CA sequence also, something like that. So two nucleotides will be repeating continuously. Or single nucleotide also, maybe a stretch of T continuously, or A continuously. So depending on that, you find out your markers. So something called there are something in SSRs, there's something called micro satellite and mini satellite. Micro is like 2 to 5 repeats will be there. Mini, it's just a classification. So mini satellites will have 5 to 9 random tandem repeats. So based on that, you can these are present in DNA, so you need to specifically identify where they are present, and you can use that for your advantage as well. So there is something called um, simple sequence repeat also. So here, as I said, like these are the simple sequence repeat. There is one stretch of CA here, and in another place there is a simple stretch of TG here. So you can use this for your advantage, and you can identify what is present in between, that is inter SSR. So between two simple sequence repeat, there is something which you can use that stretch as well for your advantage. This is one more method. And then the most recent and the most employed method is SNP, that is single nucleotide polymorphism. What is this in the sense? So if you see among individuals at the same position of the DNA, there will be a difference in one nucleotide, that is single nucleotide difference. Okay, so this individual has at the see all other nucleotides are same except for this A here for this individual at this position, and for the second individual there is a G, and for third individual there is a T. Okay, so you identify this difference. So you do some experiments and you find out that there is a difference at this position, and you employ this knowledge for your advantage. Okay, um, uh, this will say for example, a tall person will have that on a DNA. If you see the stretch of DNA that codes for tallness of a person, so tallness is always maybe A. The person at this position who has A may be always tall, and the person at this position may be always dwarf. Okay, so you employ that knowledge for your advantage. So what do, you design your primers to identify this difference, and you do your PCR, and wherever the A is present, you can just say that person is will be tall, and the other G will, you can say that that person will be dwarf, something like that. Okay, and uh, this advantage you can always this knowledge is you can always employ to identify say, as I said, a disease. And I'll give you some more examples where it is employed in plant breeding as well. Okay, so there are something these markers is, how do you design your markers is like this is a stretch of DNA. So this green portion may be your gene. Okay, you don't have to find out that marker exactly on your gene. So there is something nearby, so there is small stretch of DNA around this your gene, like these red flags, this is where you can use, you can always see for these differences in and around this gene as well, and you can use this for the advantage. Okay. This is one more example of SNP. Okay, you don't have to worry about it. Okay, so when do you decide like which method you want to employ for what kind of detection? Okay, so this is a table about that. So RFLP, RAPD, AFLP, SSR, and SNPs, which we talked about. So there are some differences in those advantage and disadvantage. Depending on that, you have to decide which method you have to follow. Okay, so you need the DNA required is a question. Okay, to do your RFLP, you need more amount of DNA. If you see here, okay, RAPD will need lesser amount because you are employing PCR here. So you take small amount of DNA and you multiply in the laboratory and then do your experiment. So you need very less amount of DNA there. And SNPs also. So wherever you are employing PCR, you may need lesser amount of DNA. And where you don't employ your PCR, you need more amount of DNA. And here the quality of the DNA also matters. So so some of the marker detection needs high quality DNA, some needs moderate quality DNA, because those kind of methods work when the DNA is not so clean also. Okay, and we saw this PCR based, which method employs PCR, which method doesn't employ PCR. And ease of use. So it is not so easy, and all these methods are easy to use. So you just do your PCR and then use your restriction enzyme and then identify differences in some cases. And SNPs, you need little more experiment to use this SNPs. Okay, and then amenable for automation. So because since we are doing experiments in very large number, we need to use it in thousands and thousands of individuals. So you need to see that whether you can automate that process as well. So there some methods are very manual and cumbersome. Some methods are very easy, and you can do it in bulk at very easy pace. So those are the ones which are like you need to see depending on your requirement or the condition. And reproducibility. So you should always reproducibility is very important in science or in your experiment. So always go with some methods where the reproducibility is very very high. And also cost matters a lot, depending on the budget you have for your experiment, the lab where you are working, and what you're trying to find out. So but depending on the cost also, you need to decide cost, and then reproducibility, amenability for automation, quality of the DNA. All this determines which method you need to employ. um i- i- am I okay? Should I pause? Are there any questions?
So far, no questions, Shobha. I think it's okay. You can go on. Are there any
Okay,
questions
how
anybody
much
want to?
time I have, Nick?
Another 5 minutes,
Okay, sure.
Shobha.
Okay,
Yeah,
yeah, thank you.
we are doing good on time. No problem. Yeah.
Okay, sure, thanks. Okay, so last two slides I have. So coming to the application, so we saw various methods of detection. So how do we use this knowledge in biotechnology? So there are some general application and then there are some specific application. So general application is like parentage determination. So you want to find out the parents of an individual or parents of plant where it is coming from. So those detection you can use these markers. So parentage detection, disease detection, sex detection. So you might have heard about determination of sex in the embryo itself. So those these are some of the methods that is employed for sex detection. And identification of carrier, like as I said, since we look into DNA and identify the differences, so some diseases, if you take human beings, some diseases don't express in your early age. So as the age progresses, some diseases start appearing in a person. So it means that you always carry that, and it is not expressed. Some of I think sickle cell anemia, and then some genetic diseases, you see that as carriers. It expresses in a homozygous condition, it doesn't express in heterozygous condition, something like that. So you can always see when it is in heterozygous condition, you still can identify the carrier. And it is a kind of genetic barcoding as well. Since you can identify the difference between individuals, you can easily say the difference in their genetics as well. And genetic diversity, so the more number of polymorphism you see between two individuals, it says that they are more distant. So it will also say how diverse they are. And same knowledge can be employed in forensic science also, when you're investigating some crime. And that this same knowledge is used in animal breeding and also crop improvement. So coming to the specific example of animal and plant breeding, so you can use that for DNA fingerprinting. It means that you can know everything about an animal or a breed or an individual. And then you marker assisted selection is one method where you employ to improve your crops. So there you use some of these genetic markers, and you select your plants to make it better. And these can be same as genetic markers. And gene map, you can map all the genes that is present on an individual based on these genetic markers. And then gene transfer, when you're trying to identify a gene and then do gene transfer, you can employ these method. And also cloning, which till I talked about in the beginning, you can identify where whether that gene is present or not, and then take it and then clone it. Okay, so these are some of the examples where these genetic markers are employed in agriculture specifically. So this is this picture shows about this portion is about conventional breeding, and then this is marker assisted breeding. So in conventional breeding, when you're crossing two plants and you get lot of seeds, when you sow that seeds, you have to wait for the seed to grow and then become a plant, and then identify the differences between those. That is like this is something like your morphological markers. Okay, so you have to wait to see those differences. But in marker assisted selection, what you see, you cross between two parents and you have the seeds, and you take a small you small portion of the seed, or you germinate that and take a small portion of the leaf, and you do this marker analysis, and you can identify specifically what markers, what plants you need when they are still small, or what which seeds have your desired trait, and then pick them up, and you can use it for your advantage. This picture shows about the difference in the difference as I mentioned here. So here they are trying to screen plants that is having disease resistance and susceptibility, identify the plants which are prone to disease, and identify the plants which are resistant to diseases. So when you run a gel and try to identify the marker, these are the patterns you see, and you identify which is resistant and which is susceptible. Okay, and another advantage of it is like conventional breeding versus genetic modification or marker assisted selection is conventional breeding. As I said, you each generation you do a cross, and then you have to wait to grow for the plant to grow completely, and then you make your selection. So that would take around 10 to 12 years for you to get what you want. Okay, here in this example they've shown about this plant has virus resistance, and this is a high yielding crop, but this doesn't have a virus resistance. So if you want this high yielding crop to have a virus resistance, you make a cross between these plants continuously for cross, and then see in the next generation which plants has both high yielding and virus resistance, and then cross them many times to get the virus resistance and high yielding crop at the end. That would take 10 to 12 years. But with this genetic modification or marker assisted selection, what you already know the DNA of it, DNA sequence of this virus resistant plant, you would have identified using some markers in the DNA. You can say that this portion of the DNA is giving virus resistance in this plant, and you also know this high yielding crop. So this crop is high yielding because of these characters you have identified. So when you make those crosses, when the plant is still small, you can make the selection, discard that which doesn't fall under your selection, and then get an virus resistance high yielding crop within 4 to 6 years. So these employing these markers will actually reduce your development of your high yielding crop or the highly desired crops. And then this is one example which I took from the publication that was there outside. So you have an these are tomato hybrids. Okay, so this one of the hybrid had already resistance for 3 diseases, and in on top of that they have used this marker assisted selection to add that this TY2 is TY2 is a virus resistant. So these are bacterial resistant, TOLCV, bacterial wilt, and early blight. These are bacterial diseases. So one of the tomato variety, your hybrid already has resistance to bacterial disease. On top of that, you are trying to put the virus disease resistance also into the same plant. So they employed this marker assisted selection to do that, to bring those virus resistant characters in these already bacterial resistant character tomato plant. Okay, and there are some examples of value addition in the crop. So uh, you find out essential amino acid in and total protein, to identify total protein, and then essential amino acids, and then to reduce anti nutrition factors like oxalate, which is an anti nutrition, and then there were and to increase shelf life of tomato and capsicum. They what scientists found out was uh, cell wall glycoproteins they had to, one of the proteins that was as the fruit ripens, right, tomato and some of the fruits like say tomato and capsicum, as the fruit ripens, there are some proteins called glycoprotein that is produced, which dissolves the cell wall. That's how they become soft when the fruits are ripe. Okay, so what scientists did was they silenced this or they they found some lines where they don't produce glycoproteins are not there. Okay, they transferred that character into normal tomato plant or capsicum plant, where so those cell wall degrading enzymes are not there. When they're not there, the shelf life of tomato and capsicum increase. So these are some of the examples of these marker assisted selection in crop breeding. Okay, and thank you all for listening. So if there are any questions, I can take.
Thanks, Shobha. I think what I'll do is I'll show them the video of our lab, virtual
Okay,
tour of our lab,
okay,
and then maybe there'd be questions on. It kind of follows up or sums up what Shobha's been talking about how we use molecular markers to speed up our breeding programs. So the lab that we have in Bangalore is a SNP SNP-based marker lab where we do a lot of data points at one time. Okay, so I'll just share that video with you all. And okay, so just a minute. Yep.
While Anik is sharing, there is a question like, will there be a recorded version for us to rewatch later? I think I can see Lauren recording, so maybe that will be circulated among participants.
Yeah. Okay, so I hope you all can hear this also.
The samples that are analyzed in the genotyping lab are leaf
Yes,
pits
Anik.
Yeah,
come in as
thanks.
fresh tissues in 96 well blocks. Each block can hold up to 96 samples, which will be analyzed together. The fresh samples are first freeze-dried by placing them in a lyophilizer overnight. After they are dried, beads are added and shaken vigorously to powder them in preparation for DNA extraction. For DNA extraction, known quantity of extraction buffer is added to each block using the 96 well head in a liquid handling system. The tips are first loaded onto the 96 well head, which then moves to a trough containing the buffer, where the buffer is first aspirated and then dispensed into the blocks that have the powdered leaf samples. This step can be repeated many times to transfer buffer to other blocks. This way, a large number of samples can be analyzed at one time in a high throughput manner. The liquid handler can be programmed to add buffer to numerous blocks at one time. Once the buffer is added, the blocks are capped and homogenized by placing in a homogenizer, and then they are ready for spinning in a centrifuge. The plant debris are separated from the extracts by spinning the blocks in a centrifuge. During centrifugation, because of the centrifugal force, the plant debris pellet at the bottom of each of the wells in the block. Equal number of blocks are spun together to ensure that they are balanced during the spin. After centrifugation, the supernatants are separated from the pellets using a liquid handler, and the DNA is precipitated in these blocks. The DNA pellet in the blocks are then resuspended in buffer by using a dispenser. The dispenser will add required amounts of buffer to all the wells in the block to get them ready for the PCR setup. In the dispenser, the blocks are first placed on a tower, then it is automatically lowered to a conveyor belt which moves it to a dispensing unit where the required amount of buffer is added to each of the wells in the block. The DNA pellets dissolve in the buffer and then they are quantified and used for the PCR setup. The PCR reaction is set up in nanoliter volumes on PCR tape. DNA is first transferred from these blocks onto the tape, after which the PCR mixture is dispensed into the wells. The Nexer is a highly sophisticated liquid handler that can add nanoliter volumes of reagents for the PCR with great accuracy and speed. Once the PCR mixture is added to the tape that has the DNA, the tape is then sealed and spooled and made ready for the PCR machine. PCR is carried out in a hot water bath with the required temperatures for the PCR reaction to occur. A robotic arm that holds a spool of tape that has the DNA and the PCR reagents are immersed in the water bath with the required temperature. It is here the DNA is first denatured, then the primers anneal to the DNA strands and they start extending. The entire PCR cycle is repeated many times to complete the PCR reaction. By carrying out the PCR reaction in a water bath, we are able to set up PCR reactions for large number of samples at one time. Once the PCR is complete, the tape is read in an arrayer. The arrayer reads the fluorescence of the PCR products in the tape, and the results appear on a plot. The plots are then scored, and the results let us know if the SNP marker that we are interested in is present in these plants that are being tested or not.
Okay, so that was an overview of the genotyping workflow that we carry out in our laboratory. Now we also do testing for biotech traits. That is, we use to check insect control activity to see how the insects respond to various toxins. So I'll also show you a video on that in a minute.
We rear different insect pests in the insect control laboratory at the Bayer Biotech Research Center in Bangalore. What you are looking at is a scientist transferring insect eggs using a paintbrush onto insect diet in the insect trays. The insects are reared in these wells with known concentrations of toxin to evaluate the most effective concentration to kill or inhibit the insect development. The larvae are periodically evaluated and placed on fresh plates as they grow. This is how we can estimate the right concentration to effectively control the insect pest. This is an insect box with the different developmental stages of the American bollworm Helicoverpa. These Petri plates have live specimens of different insects that are reared in the lab. There are 4 Petri plates for each of the insect pests, one with the insect eggs, the other the adult insect, the pupa, and the last one have the damaging stages of the insect, which are the caterpillars. This insect box has different developmental stages of the tobacco cutworm Spodoptera littera. The vials have different stages or instars of the pest along with the pupa and the adult moths. These are the live specimens of Spodoptera and Ireus. The larva is what damages the crops. They are the pupa, the adult moth, and eggs. Here are the eggs of Pectinophora and Helicoverpa, the adult moths, the pupa, and the damaging stage, the larva. These are the various stages of the pest Chylopatulus. And these are the different stages of the American bollworm. The eggs, the 6 instars, left to right, the pupa, and the adult moths. We rear different insect pests in the insect.
Okay, so that's what we had for you guys. So we are open to any questions. We have a few more minutes where we could take any questions if there are any. You could either send them as chat or unmute yourselves and ask us the questions. We'll be able to answer.
Well, the problem is that I can't offer some very complex questions because I don't have any knowledge in genetics or stuff like that. I only came here out of curiosity and maybe hopefully I could learn something from it. I don't understand the whole more precise genetical stuff, but I can ask if genetical engineering will be used on humans in the future.
Okay, so as far as my knowledge goes, they have not, there's no approval from any country to do any kind of genetic engineering work on human beings per se, because you could be altering the DNA of human beings and kind of making, I mean, it could be not used in the right manner. Okay, but there are other technologies that are coming up, what they call gene editing, that is a little different from this, where it could be used in therapeutics in order to cure cancer and some
It is
genetic
more
disorder.
present, it's more immediate for the people.
Correct.
they could get to see the benefits right away.
Yes. So that technology that I was talking about is now still in the testing stage, but people are kind of getting more aware of it and trying to see how they could probably start using them in the medical field. I'm not sure if you are aware, but just about a couple of months ago, the Nobel Prize was given up to two women scientists in chemistry who discovered this whole gene editing or the CRISPR
I
technology.
know.
Yeah. So this is mostly in that area. Yeah. So thank you for the question. Is there anything else?
I don't, to be honest. A lot of stuff is complicated that flies over my head, but
Okay.
I understand a couple of things. I mean, I
Yeah.
just say if I want to say that I had a teacher at my university who just he was negative about the genetically modified organisms.
Okay.
I don't remember in full, but it's mostly about the Monsanto scandals and the whole hormonal problem. I mean, basically, he said that people who ate genetically modified foods had hormonal problems and disease. I mean, he said,
Y- Yeah, so just for your information, I mean, genetically engineered crops have been grown for more than 20 years now. So if that is really the case, we should have been seeing a lot of damaging things happening to people. But on the contrary, if you see in a country like ours in India, it's revolutionized or changed the whole scenario of the farming community, where here it's cotton that they're growing, but India becoming a major exporter of cotton from importing. Okay, so as I was mentioning, all technologies can be misused, but no regulators would allow them, any company or an institute, to commercialize any product that is going to be harmful to the environment. So and they undergo a lot of rigorous testings. So
I understand. I'm not saying
yeah,
I'm not negative about the whole thing. I'm just saying what
you
other people. I mean, quote unquote academically qualified people, supposedly.
correct, yeah. So that is another initiative uh, many people are taking to speak about the technology so that they understand it better. Okay, so
Yes.
yeah, there's one question over here. Yeah, is there a technology working on increasing the oil content in oil crops? So as Thilai was mentioning, and even Shobha, like there are different ways you could increase the oil in an oil crop. Now, if you want to use a marker technology where you just associate a molecular marker with higher oil yields in a certain crop, you can kind of use the marker technology to incorporate that gene or that marker into other crops. Or you can use technologies like transgenics, where you can identify a gene that can yield higher oil from one crop and put it into another crop. So that's an answer to the question on increasing oil content in oil crops. So it has been done, and there are quite a few crops that people are working on, like in canola, in to increase the oil content, then camelina and other crops. Yeah. Are there any other questions? Yeah. Could people create their own genes from scratch? I don't think we've reached a stage uh, of that as of now. So the question is, could people create their own genes from scratch?
Um, Also, is it possible for the genetically modified organisms to to escape the whole the area they're being grown in and mixing with the wild environment?
So once a genetically modified crop is produced, So usually the regulators and the institute who make it have all these experiments where where they need to prove that the gene will not escape into the environment. So if they are scared, it could cross-pollinate with other related species and the gene get transferred where they do not want it to go. So usually all these studies are pretty much taken care of. So that is another reason why, like Thilay was mentioning in the first presentation, the time involved in developing these crops, because they have to undergo all these rigorous treatment, rigorous tests before it can be set out. So usually all these are taken care of before it could be commercialized. If these experiments are not satisfactory, the regulators in the countries will not let them be commercialized. I hope I
Mm,
answered your question. Yeah,
thank you.
you're most welcome. Is there anyone else who have any questions? Okay, so there were a couple of questions in the that had come up before the meeting. So one was on glyphosate. So Shubha, would you like to take that? So the question was on how do we handle the glyphosate saga, the lawsuits, and do they really farm the farmers?
Okay. Um, so talk about glyphosate, glyphosate is actually a very safe chemical. The proof for that is like it has been tested for, that is the most tested chemical that is there existing till now, because glyphosate is around for 40 years. So that itself will say that like it is a very um, potent chemical that the world needs, and till now there is no replacement for glyphosate uh, as an herbicide. Um, to add to that, like glyphosate can help the environment by keeping the soils healthy, because when you're spraying the herbicide, you are less disturbing the soil, and then organic, when soil is less disturbed, it will develop good quality, like more of organic matter, and soil health increases, and soil moisture increases, and soil erosion decreases. To add to that, like glyphosate, uh, with 40 years each year, um, US Environment Protection Agency, as Anik was mentioned, there is regulating authority who who looks at the safety of all the chemicals. So out of that, US Environment Protection Agency, European Food Safety Authority regulators, and regulators in Canada, regulators in Japan, regulators in Australia, Korea, Brazil, or all of them review and approve these pesticide products, and they are also monitored year on year. So glyphosate has been certified for 40 years that it is safe by all the regulators. So there is no risk, uh, no risk to public health using glyphosate. So coming to a lawsuit, um, um, okay, some people who have filed the lawsuit for glyphosate are mostly not farmers. Farmers use this glyphosate extensively for controlling weeds, but people who have filed lawsuits are from are the home people who use them in lawns and gardens. So their use for glyphosate is very less. And then exposure to glyphosate is also very less. So that says that their intention of filing the lawsuit was different, or something that was not didn't have a um, good intention to. Okay, and then what happened after that, like because the lawyers gave wide publicity in the media and everywhere on these lawsuits, more and more people filed lawsuits on glyphosate. Coming to what company did, so it has put out all the documents that is there about safe use of glyphosate. So Bayer for Bayer on follows that being transparent is very, very important, and you can go and check on Bayer website about the glyphosate. Bayer has shared 107 safety studies as part of the transparency initiative. So you can find all those documents on Bayer website. So 107 safety studies has been done before year on year and before release of glyphosate to public use. And then you also find 100 Environment Protection Agency studies on cancer risk analysis for glyphosate, and more than 800 safety studies submitted to the regulators on glyphosate. And in 2018 also there is an independent studies by um, a National Institute of Cancer supported by Agriculture Health Studies on glyphosate. All of them have proved that glyphosate is safe. Okay, so what company did to settle all these glyphosate issues is like it since this glyphosate lawsuits were dragging so many years, and it was creating uncertainty in the company and in the business. And some countries what they did was, till these lawsuits are settled or there's a final verdict, we will not allow glyphosate to use. That was like some countries took that decision, so that was also affecting the business. So what Bayer as a company did was, it came with an agreement, mutual agreement or mutual settlement with the suitors, so that to put an end to this uncertainty and to continue with the business. So with that, like the freedom to operate for glyphosate is there. And glyphosate is since there are no reports about no scientific studies or reports stating that glyphosate causes cancer, so glyphosate is safe to use is what regulators have certified. And then we have freedom to operate and use and sell glyphosate everywhere, and that also gives certainty to the company and some direction, so that the company will look focus on other research activities. So that's what the glyphosate story is till now, and till now there is not a single document or a single proof to say that glyphosate is unsafe for use. So any thing Anik or Tillai can
Yeah.
add to that if
No,
I
Shobha.
missed any something.
Yeah, I think you covered it all. And the fact that it has not been, the regulators have not asked us to take it off the shelf or stop selling it also proves that it is a safe chemical. That's why it's still continued to be sold. Yeah.
Yeah. True, Shobha. You have covered everything, Shobha. And one more thing to add is again, the half-life of glyphosate is 2 to 45 days. So after 45 days, it just again degrades and it's turned out to be a carbon source. So that's one more thing.
Okay.
Yeah,
Yeah,
thanks
important
Tillai and
point.
Anik.
Thanks, Tillai. Yeah. Okay, so I think we've gone 4 minutes over, so we better wind up. So thank you, Shakti and Laureen, for this opportunity. And we'd always be out here. So if you all want to touch base and understand more about science, you can always talk to us through these, either through them or drop us an email. Yeah, so Shakti and Laureen, can we log off?
Yes, thank you everyone so much for coming.
Thank you.
It was great having you. We will be
Yes.
recording this and sending it out. So
Yeah,
thank
Thank
you.
you.
thank you.